Now, structure of microsporangium. This is the cross section of anther, which is roughly tetragonal or four-sided, and these are the four microsporangia. The microsporangia are circular in outline. Each microsporangium is surrounded by four wall layers. There are four wall layers which cover the microsporangia. The outermost is called epidermis. Inner to epidermis lies endothekium. Inner to endothekium lies middle layer. Epidermis outermost. Inner to epidermis endothekium. Inner to endothekium middle layer, and the innermost is the tapetum. Epidermis, endothekium, middle layer and tapetum. It is clear here? Epidermis, endothekium, middle layer, tapetum. Tapetum is the innermost wall layer. Now, epidermis, endothekium and middle layer, these three outer layers provide protection and help in dehiscence of anther. They have the same function. They provide protection to the anther and help in the dehiscence. But tapetum has a different role. It is the innermost wall layer which consists of very small cells. The cells have dense cytoplasm and more than one nucleus. Tapetum consists of cells with dense cytoplasm and more than one nucleus. Tapetum provides nutrition to the developing microspores. The tapetum provides protection to the developing microspores or pollen grains because the cells of tapetum secrete enzymes, hormones, and many other nutrients. So tapetum helps in providing nutrition to the developing pollens or microspores. These are the microspores which are developing. So they receive nutrition from the innermost layer that is called tapetum. Now, this is about the wall layers of the microsporangia. Now, when the anther is young and immature, it is not ready for dehiscence. Inside each microsporangia, the center of each microsporangium is occupied by a tissue which is called sporogenous tissue. Sporogenous tissue is a cluster of compactly packed homogeneous cells. Sporogenous tissue is a cluster of homogeneous cells which are compactly packed, and each cell is capable of developing a pollen grain. So sporogenous tissue occupies the center of each microsporangium. Sporogenous tissue is a group of compactly packed homogeneous cells. Each cell of sporogenous tissue is called pollen mother cell or microspore mother cell. Sporogenous tissue, it is a group of compactly packed homogeneous cells. Each cell of sporogenous tissue is called pollen mother cell or microspore mother cell because each cell has the potential to give rise to 4 microspores. This is the microspore mother cell. Each microspore mother cell is a part of the sporogenous tissue, and each microspore mother cell can give rise to 4 microspores through meiosis. Meiosis results in the formation of 4 daughter cells. It is a reduction division. The pollen mother cell or microspore mother cell has the ability to give rise to 4 microspores through meiosis. So formation of microspores from microspore mother cell through meiosis is called microsporogenesis. Microsporogenesis. The 4 microspores remain clustered in a group of 4. This is called microspore tetrad. The 4 microspores which develop through meiosis remain attached. They remain clustered, forming microspore tetrad. Tetrad means a group of 4 cells. So from each microspore mother cells, microspore tetrad develops. When the anther matures, dehydrates, the microspores dissociate, they detach, they separate from each other, and they develop into mature pollen grains. So microspores develop into pollen grains. The microspores remain in a group of 4 in the beginning, which is called tetrad. When the anther becomes mature, dehydrates, the microspores detach, they dissociate from each other, develop into mature pollen grains. So in this way, the pollen grains develop. The whole process is called microsporogenesis. And the tissue, the original tissue from where the pollens develop, is called sporogenous tissue, where each cell is a potential microspore mother cell, and each cell is a diploid cell. Because of meiosis, they divide into 4 haploid microspores. So microspores or pollen grains are haploid. Pollen mother cells or microspore mother cells are diploid. A diploid cell undergoes meiosis to form 4 haploid daughter cells. This is meiosis. So microspore mother cells are diploid, microspores are haploid, pollens are haploid. So in this way, thousands of pollens develop within the 4 microsporangia. The process is called microsporogenesis. And when the anther becomes mature, the anther wall dehisces. The wall of the anther ruptures along the line of dehiscence to release the pollen grains to the external environment. This is the dehiscence. Pollens are released through the line of dehiscence as the anther wall ruptures when the anther becomes mature.